Thank you. So I'm an associate professor in gerontology at the University of Southern California. So I'm very interested in aging and aging-related diseases. And more recently, I've become interested in how mental illness can be a risk factor for neurodegenerative disease. And there has been some research on that to the effect that psychiatric disease is a risk factor for dementia. And there have been a number of meta-analyses and epidemiological studies to situate psychiatric disease, anxiety, and depression in particular, relative to dementia as the outcome factor in this set of comorbidities that may shorten lifespan. And as it turns out, in meta-analysis, as you can see on the slide, there's a 29% high risk for Alzheimer's disease in meta-analysis for Alzheimer's. So there's a 29% high risk for dementia, especially Alzheimer's disease, in persons who have clinically significant anxiety. And there have been similar studies done to evaluate the relative risk of dementia in persons with depression. And the findings have been similar, which is, that, for example, that for vascular dementia, the risk is even higher, about an odds ratio of 2 relative to healthy controls without anxiety. And so there seems to be an increased risk for Alzheimer's disease and vascular dementia in persons who have a history of anxiety or depression. And the reason this is important, I believe, is because it's important to find people who are at high dementia risk early to increase the window of opportunity for interventions that might delay the onset of the disease, or that might even identify persons who are at high risk for early monitoring, early intervention, and even to stratify them for treatment using some of the drugs that were recently approved for Alzheimer's. Unfortunately, one challenge is that models of Alzheimer's risk do not generalize well across populations, across ancestries, and even within more homogeneous samples of similar ancestry, there is still a lot of work to be done to improve models of risk. And so one of the purposes of my lab is to optimize these models and to create new models that can assist in identifying people at high risk for neurodegenerative disease and dementia. There is in Middle Eastern and North African countries a sharp expected spike in dementia risk according to epidemiologists from the World Health Organization. And these populations are currently underrepresented in studies. And so I'm very glad to be here and to see this wonderful research being done in Turkey to expand the scope of neuropsychiatric studies across different ancestries. There is a plot I have here in the bottom right corner showing how the prevalence of the ApoE4 allele, which is one of the strongest genetic risk factors for Alzheimer's disease, differs significantly between European populations like the British or Germany and countries in the Middle East and North Africa. And so there is a lot of heterogeneity in risk for Alzheimer's that hasn't been accounted for. And so it's important to understand these factors and to understand how psychiatric disease is a risk factor for Alzheimer's, which is something that hasn't been studied very much recently. There is a long literature showing how emotional stress has effects on neuronal morphology. So for example, in these data on the bottom left, you can see how in stressed mice, neurons have different morphologies. So in anterior cingulate cortex and in the hippocampus, the neurons have fewer synaptic spines in mice that are subjected to chronic stress. And these findings are correlated with MRI findings on atrophy, specifically gray matter volume loss, seems to be associated with these morphological changes in neurons that have been documented. And you can see in the middle plot, the blue is the number of dendritic spines for mice that are not subjected to a stressful environment, and the red are the mice that have been subjected to stress. And you can see that the number of dendritic, the number of dendrites, the dendrite density is much lower, and so there are these morphological changes that occur that can be detected using imaging techniques such as magnetic resonance imaging. And there are differences across regions, across conditions, and it is a dose-dependent effect of stress, which we believe is correlated with anxiety in humans across the spectrum of different exposures to stress. We do know a lot about how anxiety and stress affects brain regions differently. So for example, the frontal lobe is sensitive to chronic stress, to alcohol exposure and to recreational drugs. The insula can become hyperconnected to the rest of the brain in anxiety disorders and depression. There are changes in the parietal lobe, which mediates mental arithmetic. So one of the easiest, simplest tasks that involve stress is actually a mental addition task. And so you can see how cortisol levels and other markers of anxiety increase in individuals when they are subjected to this mental arithmetic task. So when you have to add or multiply two numbers in your head, your cortisol increases and your skin conductance response is also higher. There are also changes in the temporal lobe and in subcortical structures, particularly the hippocampus. And there is abnormal connectivity that has been mapped using magnetic resonance imaging and diffusion tensor imaging. And so there is really a wide range of changes in the brain that occur as a function of stress severity and duration. And so chronic stress is, for that reason, thought to lead to a high risk for Alzheimer's disease. What's interesting, though, is that even though this link has been acknowledged, there is not a lot of work in understanding how we could use information on anxiety or depression in individuals to improve our estimates of dementia risk. And right now there is a situation where healthcare systems are really poorly equipped to leverage expertise from psychiatry and to monitor and to understand dementia risk in view of reducing that risk. And so if you look at the 12 risk factors for dementia, you can see social isolation and depression being listed. But a lot of the other risk factors are often comorbid with both of these. And importantly, anxiety is not yet acknowledged very broadly as a risk factor. And so to reduce risk for dementia, usually individuals are recommended to make lifestyle changes. But there is an underappreciated role of psychiatric treatment and monitoring of persons who at highest levels of anxiety or depression to better estimate their risk for Alzheimer's disease. So to estimate that risk, my lab uses magnetic resonance imaging to quantify the biological age of the brain. So if you think about it, if the brain is aging faster than expected, the risk for Alzheimer's is higher. So that has been documented. For example, in persons with Alzheimer's, the average biological age is about 10 years older than in healthy controls who do not have cognitive impairment. There's evidence, as you saw from the animal models, that the chronic stress that these animals are subjected to increases oxidative stress and leads to changes in the morphology of neurons. And so there is a fair argument to be made that perhaps psychiatric disease is associated with a higher rate of biological aging in the brain. And so to go further into that, I'd like to first explain how that works. So if we look at a group of individuals of a certain chronological age, so chronological age is the age that essentially we think of when we're thinking of age. So you see in the top right corner, I have that plot of chronological age versus biological age. So if we take a large sample of individuals of the same chronological age, and we estimate their biological age from measures such as DNA methylation, we find that the results follow a normal distribution. So subjects in this large sample of individuals of the same chronological age have a distribution of biological ages, and about half of them are biologically younger than 38, and about half are biologically older. And so we feel that perhaps the individuals who are biologically older than they should be could be at higher risk for Alzheimer's disease, for example. And so this is an example that explains very well how biological age works. So if you have a person of chronological age 35 and they have a biological age of 40, that's evidence of past accelerated aging and perhaps shorter lifespan or health span. By contrast, if you're at age 35 and you have a biological age of 30, then that's younger, and so you expect a longer health span and perhaps lower risk for neurodegenerative disease. And so what's important to know is that chronological age does not capture this variability in aging very well. If you plot chronological age on the x-axis and biological age on the y-axis, you have the line x equals y on either of these plots that show typical aging. But anybody who falls outside that plot is experiencing acceleration of aging. And individuals who are below the line x equals y for normal aging are experiencing perhaps deceleration. So both of these groups are of interest because people who are aging slower than expected may help us to understand factors that can assist with resilience to neurodegenerative disease. And those who have aging acceleration can help us to understand what kind of lifestyle factors or genetic factors can increase the risk for Alzheimer's disease. So my lab has developed some tools to estimate brain age from MRI using deep neural networks. And these are computer models that estimate brain age from MRI. And the way they work is that they take as input an image. So in this case, you see the image of a cat on the left. And this image is passed on to a series of blocks, as you can see. There are different blocks that do different kinds of operations on the data to identify patterns in the images. So this is an example of a convolutional net that analyzes brain images to find patterns that reflect aging. So if you wanted to estimate the age of the brain using convolutional nets, you can replace the image of the cat with MRI volume of the brain, and then these images are processed, they go through each block, and at each block you look at different patterns in the data that can help a neural network to estimate age. And so you get to the very last layer and on the very far right, and then you get an estimate of the age. And so essentially what you're doing is convolving the image with these filters that allow us to capture different patterns in the data. And on the very far left, you have filters at a relatively small spatial scale. As the data is passed along the blocks, you're starting to look at larger and larger physical scales because you're also collapsing across the dimensions of the data. You do some averaging across the voxels in the volume, and so you're collapsing across and decimating the volume, but which also actually lets you look at larger spatial scales. So the convolutional neural network looks across all these scales, both small and large, and identifies patterns that help it to optimize on its predicted value of age. So this is a procedure illustrated here, and it's taken from a neural network whose purpose is to classify dogs according to their breed. So if you wanted to classify a picture of a dog and identify the breed of the dog, you can break down all the pixels in the image as shown in this figure, and you get all the way to the top where you have those very small images, and those are the most fundamental patterns, the most basic patterns in the image. And then as you go through different layers, which are horizontal rows in this image, you're looking at ever larger scale of the data until you start looking at very large scale features of the animal in the image. And so that allows you to essentially pool across all scales of features to identify which features are most useful for discriminating between a dog of a certain breed and a dog of some other breed. So what we do with these neural networks is very similar, except that the input is an MRI that is looked at by the neural network to identify patterns of aging that allow the neural network to determine the biological age of the person. So we trained, in our most recent publication, a neural net to predict brain age. This was trained on subjects without any neurological or psychiatric disease, because this is required to accurately predict age and calibrate a neural network according to the most salient features of aging and for a certain age group. And so we pooled across data in several large repositories, including the UK Biobank, and we also looked at the CAMCAN sample in England, and the ADNI repository in the United States. The Human Connectome Project also was included, and more recently we've also included other data sets to improve generalizability. And so it's very useful to include as many data sources as possible, because that helps to prevent overfitting and to help with the generalizability of the neural networks. So we assigned participants into training and test sets of sizes equal to about 80% for training and 20% for testing, and initially trained on about 5,800 people to predict brain age. And one innovation we had in this context was using these saliency maps to actually identify anatomic locations that are more salient to the network. So one big problem with neural networks is that they are black boxes. So because they're so complicated, it's very difficult to understand how neural networks achieve their purposes and their aims when they try to estimate age. So there are several methods that have been proposed to provide interpretability to neural networks. One of them is probing, which looks at the inner workings of a model to understand how important a certain feature is to the determination of age. And this is a very time-consuming process, because you have a very large number of computational units in a network, and so it can be very difficult to understand, to gain biological insight from this kind of operation. Other methods that have been proposed are perturbations. So if you change something in the input data, you can look at the effect of that change on the output, and that can tell you something about the importance of a feature. So this is what's done in occlusion mapping, for example. And it's also a very tedious procedure, because if the data are very complex and you have to perturb every small element in it, it can take a very long time to understand how the different features are contributing to the output. Another way to do this is using surrogate data. So if you create your own data with certain features, you could pass it to the neural network and understand a little bit about what's going on inside. But usually these are all very computationally expensive ways to understand how a neural network works. So we adopted this approach called saliency mapping, where you're looking at how individual voxels affect the weights of the neural network at a global scale across all the layers and blocks of the neural network. And so there are a number of these methods in current use. Some are more useful than others. You see here in the case of the dog and cat, each column shows the saliency map of a neural network according to the saliency methods results. And you can see that some saliency methods focus on one region more than another. Some are more broadly distributed spatially. Some have more focal aim. And so it's important to compare these methods for saliency mapping and understand which ones provide the best biological insight. And so we did that. We built a simulator where we actually manipulated features of the input MRI to see whether the neural network could find these changes. So in this example, we took one small region in the prefrontal cortex and added just one layer of gray matter. So we replaced the intensities in the white matter with the intensities from the adjacent gray matter voxels, and essentially inflated the gray matter label into the white matter. And so if you do that as a function of age, in other words, if you increase the number of voxels by which you grow the gray matter as a function of age, then that feature will be correlated with the age of the patient. And so the neural network should pick up on that if it's doing what it's supposed to and identify that region of the brain as having high saliency. And so we did this, and the result was very encouraging. So we found exactly that kind of saliency being concentrated around the region where we only changed the image by one voxel layer in the precentral cortex. And so this method goes through and identifies also regional patterns of aging that are specific to individuals. So for example, at the bottom, you can see that the input is the MRI from the skull stripped MRI from various subjects. And the subject saliency map is created to indicate which parts of the brain are most salient to determining brain age in that particular subject. And so you can average the saliency maps and look at differences between groups. We looked at differences in aging across males and females. You can also look at differences in any kind of statistical comparison between groups that might be of interest in a study. Another encouraging step that we had relative to finding the importance of and the utility of our saliency mapping involved comparison of saliency differences between age groups and differences in cortical thinning. So the part D at the top left shows differences in cortical thickness between age groups. So we compared individuals in their 50s, mid-50s to mid-60s to individuals in their mid-60s to mid-70s. So we compared and we averaged the cortical thickness across these two groups and then took the difference. So these maps at the top right show differences in cortical thickness between these age groups. So it's a map of cortical thinning. And so and then at the bottom on the right, you have age differences in saliency. And so we did the same procedure as we did above, but we compared saliency instead of thickness. And you can see that there is a lot of similarity. You have a lot of saliency in locations where the cortex is thinning a lot faster, such as in medial occipital areas, in limbic areas, and anteroventral medial frontal cortex. You also have some correspondence on the lateral side of the cortex. And so we did a statistical analysis to look at how whether saliency explains any of the morphometric measures that are used currently in the field to characterize aging. So this is cortical thinning, volume loss, curvature changes, or cortical area changes. So these are the standard measures output by FreeSurfer software that have been used to characterize change with age. And so we found that the saliency has very good agreement and is able to capture a lot of these metrics and potentially also identify novel patterns of aging that are not characterized by any of these measures of atrophy. And so that's an important advantage of neural networks. You can find patterns of aging that are unapparent to humans, and whereas cortical thinning and volume loss are measures that are defined a priori to characterize aging, the neural network can identify its own complex measures that perhaps we cannot identify. So we benchmarked and evaluated this using mean absolute error, which measures the accuracy of brain age estimation in cognitively normal adults. And we found that our solution is actually has a lot less error on average than most other measures of error that other methods have. So if you do a standard regression on brain features, you actually get an average MAE of about 6 years. And our method published earlier this year achieved an error of about 2 years. And since then, we have trained a more robust version of the network on 28,000 people, and we found that actually we were able to reduce the MAE even further to about 1.4 or 1.5 years. Obviously, this needs also generalizability across samples, which is something we're still working on. But the method is, compared to others in the literature, a lot better at explaining variance in the data. As you can see at the bottom right, you can actually explain more variance than other methods can. And it has fewer trainable parameters, which is important because more parameters usually implies a high risk of overfitting the data. So these are all encouraging features of our method that others do not necessarily have or have not been characterized. Another important component of this is describing the extent to which brain age can capture measures of cognitive functioning. And so you can see here in this figure a comparison of biological age in green and chronological age in red. And on the very left, you have a comparison of the two in healthy controls. And you see that they do relatively about as well. There's not usually a significant difference between biological age and chronological age in healthy controls, which is what you'd expect, because on average in controls you have very similar biological ages. So the biological age is very similar to the chronological age on average. And so the correlation with any of these measures of cognitive functioning is about the same. But if you look at patients with Alzheimer's disease or with MCI in the middle, you see that the biological age actually captures a lot more variance and is far more strongly associated with most of the measures of cognitive function that we examined. And it's also more strongly associated with measures of neurocognitive function that are used for diagnosis of Alzheimer's disease. So this kind of shows the utility of brain age as a better indicator of risk for disease and of neurocognitive correlates of disease in this population. It's also very interesting to look at differences between sexes and groups. As you know, Alzheimer's disease risk differs a lot according to sex. And the saliency maps can help us map that. And you see in the middle, in males, you actually have a lot of saliency in the ventricles, and in females, you have more saliency in the gray matter. And these are findings that were actually already known from other studies, and our saliency framework actually captures these and synthesizes them in this nice picture. We also find that the difference between biological and chronological age, which is known as age gap, shown on the left, actually has a lower variance using our method due to lower errors in brain age estimation. And so on the left, if you see the different samples that we estimated brain age for, we looked at the UK Biobank and CAMCAN as two healthy control samples. And we find that the measure does similarly well on the UK Biobank, but on the CAMCAN, our method does better than the method with the next best error that has been published. And in the MCI group and AD group, actually, there is a significant difference in age gap from 0. So, and so the mean age gap is significantly larger than expected according to the null hypothesis. And these findings are also reproduced across the sexes. So females at the top and males at the bottom. You have a very good reflection of how biological age is actually a better able to capture the variability in the sample, especially in MCI and AD, a lot better than the other best method, which is another type of neural network that had the best documented error next to ours. And then it's also nice to look at sex differences in saliency and compare those to thickness. And as on the previous slide, you can see on the very right that sex differences in thickness are recapitulated to some extent by sex differences in saliency. And so you see these areas in the occipital lobe and in the limbic areas that are experiencing faster cortical thinning according to sex. And in the saliency, you also find these areas as being able to capture that kind of aging. However, there are also differences, and so the saliency does not is not a 100% accurate map to explain thickness, in which case it wouldn't really be helpful, but it really synergizes over thickness and other features of aging that we know and are able to characterize using existing methods. When we look at cognitively normal adults versus Alzheimer's disease, we find that these spatial patterns of aging also recapitulate findings on brain atrophy in Alzheimer's. So you can see that there is a lot of saliency on the medial aspect of the brain in entorhinal, parahippocampal areas, and in the limbic lobe on the medial aspect. And so these are all features of Alzheimer's that have been identified with other morphometry studies. And so this is further suggestion of saliency being able to capture these differences between Alzheimer's and cognitively normal aging pertaining to morphometry features. It's also interesting how white matter aging differs with sex and diagnosis. So in males, you can see on the left, you have a lot more aging, sex-related aging in white matter, deep white matter areas. In females, on the other hand, you have a more salient aging-related feature in the superficial white matter. And there's also an interesting pattern we can see on the right-hand side pertaining to the difference between cognitive impairment and cognitively normal adults. And you can see there how you have a lot of saliency in the spinal cord, in the brainstem, and in the cerebellum. And there's some evidence, we're working on a study now, suggesting how these differences in saliency might actually reflect differences in neurotransmission that are captured by other types of MRI, other than T1 weighted MRI. So in conclusion, psychiatric disease is an underappreciated risk factor and contributor to dementia risk. And if we monitor brain aging in patients with psychiatric disease, that might help us to understand Alzheimer's disease risk a little bit better and to stratify patients according to their risk. MRI can offer ways to map brain aging in persons with anxiety, depression, and other psychiatric conditions, and you can use deep learning methods like the ones I showed to map and monitor brain aging. In conclusion, I think psychiatrists and neurologists, neuroradiologists, clinicians working in preventive medicine have an opportunity to join forces to reduce the welfare burden of dementia by monitoring brain aging in older adults who are at high risk for Alzheimer's, and these methods can help with that. I'd like to thank the organizers of the Congress for inviting me, Ali in particular, and Paul Thompson for facilitating my presence, and I'm very thankful to the luxury resort we are in and for their hospitality. I'm grateful to my team and to funding we received from the National Institute on Aging. And our paper was published earlier this year. I'm happy to share it, and our code is available online. And we're looking forward to any collaborators who are interested in using it. Thank you very much.